This is a video from the topic B2 organisation, and today we're looking at enzymes. I just want to confirm a couple of definitions before we start talking about enzymes: digestion and absorption. Digestion is the role of the enzymes breaking those large insoluble molecules into small soluble molecules, and the purpose for this is so that they are small enough and soluble to be able to be absorbed through the wall of the small intestine. So that's where the word absorption comes in. So absorption is the process of those small molecules, soluble molecules, moving from the small intestine through the villi into the blood. Okay, let's not get confused between these two words, which quite often happens. Digestion is breaking down of the large molecules using enzymes. Absorption is those small molecules moving into the blood. Okay, this is just a bit of interest really. Why do flies vomit on their food before they eat it? Basically, they can't chew food. They vomit on their food to release digestive juices, enzymes and saliva, and they begin to break down the food, digest the food, and then they can suck it up as a liquid form so that it's easier for them to pass it through their body. Here we have a diagram of an enzyme. The green section is the enzyme, and the food that's going to enter that enzyme, whether it's a protein, starch or fat, starch being a carbohydrate, is known as the substrate. So the substrate fits into the enzyme. The enzyme could be a protease, a carbohydrate or a lipase enzyme, breaking down protein, carbohydrate and fat. And it fits in at a place called the active site, and you can see that actually this substrate won't fit into this enzyme because the shape of the substrate is not complementary to the shape of the enzyme. Okay, we call this the lock and key model or the lock and key theory, and it's this idea that the substrate must be specific to the enzyme. Okay, they are specific to each other. So they must fit into the active site, and we're referring to the enzyme as the lock and the substrate as the key. Okay, so if they are not complementary in shape, then the enzyme can't break down that particular substrate. So this could be that the enzyme is a protease enzyme and the substrate could be some starch. Okay, they're not complementary. So we call that the lock and key model. When we look here, we can see picture 1, we've got the active site clearly labelled. Picture 2, we can see that a substrate is approaching the active site and it's not a complementary shape, so it won't fit. Picture 3, we're back to the same enzyme. Picture 4, we've now got a substrate that is the appropriate shape. It's the right key for the lock of the enzyme. Okay, so we've now got an enzyme reactant complex. Okay, the substrate is fitted into the enzyme. And then picture 5, you can see that the products have been made. So there's been some bonds broken in the substrate, and the two products are then removing from it. So it could be that in picture 4 that was some starch, and in picture 5 it's broken down and we've got some glucose. And then just to go back and say there that picture 6, the enzyme is ready to repeat the process. So the enzyme doesn't get used up. Okay, it can then accept some more substrate and do the process again. Here is our enzyme summary, and we've already said in the digestive system video that fibre isn't absorbed by the body, it just bulks up the food to help everything flow through the digestive system. Vitamins and minerals are already small and soluble, so they don't need to be digested, and so it leaves us with the carbohydrates, the protein and the fats. Okay, so we've got 3 groups of enzyme: the carbohydrates, the protease and the lipase. Okay, let's have a look just through carbohydrates first. So an example of carbohydrates which you've probably heard of is amylase. Amylase is produced in the salivary gland and works in the mouth. It breaks the large insoluble molecule that is starch into the small soluble molecule glucose. They're both carbohydrates. And carbohydrates is made in the small intestine, the pancreas, and also the salivary gland. It's the only one that's made in the salivary gland. Protease next. Two examples here because breaking down a protein is quite complicated. We'll look at some graphs shortly showing about optimum conditions. But basically, if you've got this long chain that is protein, and you've got two enzymes. There's one enzyme called pepsin, which works in the stomach, works really well in acidic conditions. And then in the small intestine, trypsin works really well in slightly alkali conditions, and it breaks down the protein further. So pepsin breaks the protein into peptides, and then the peptides get broken down into amino acids by the small intestine. At a basic level, we just say that protein is broken into amino acids. But in actual fact, there's kind of an in-between stage where the protein becomes a peptide broken down by pepsin, and then the trypsin breaks it down further into amino acids. Again, made in the small intestine, the pancreas, but also the stomach, okay, because the pepsin's working in the stomach. Moving on to the third group then, lipase. There isn't a particular example of lipase that we need to know, but the large insoluble molecule is fat, and this time we have two products, okay? The small soluble molecules are fatty acids, which isn't too bad to memorize, and then glycerol. Watch out for that glycerol because that's one that people find hard to remember. And the lipase enzymes are made in the small intestine and the pancreas. If in doubt, always say small intestine and pancreas, because all of the enzymes are made in both of those two organs, the small intestine and the pancreas. Just be careful though, because food does not enter the pancreas, okay? So if the question is asking where are enzymes made, they're made in the small intestine and the pancreas, but where are enzymes used? They're not used in the pancreas, they're used in the small intestine. Okay. Also, the enzymes aren't used in the salivary gland, they're just made there and then they go into the mouth. Okay, so that's our summary of the three categories of enzymes, where they're made, the large molecule and the small molecule they become, and some examples. Okay, activation energy. Hopefully you'll have heard this in chemistry. Basically, particles sometimes can bump into each other with enough energy that they react. Sometimes particles don't have enough energy, and when they bump into each other, they just bounce off each other, okay? The minimum amount of energy particles need in order to react together is called the activation energy. And what enzymes do, they're biological catalysts, so they work in the same way as catalysts do when you discuss them in chemistry, is they reduce the activation energy. So that means that particles that would have bounced off each other now have a suitable activation energy, enough energy in order to react. So it means that some of those reactions that wouldn't have happened, some of those collisions that wouldn't have resulted in a reaction, actually now do result in a reaction, speeding up the whole process. So let's have a look at that as a graph. So we've got our activation energy represented by the blue line. The higher line of the two is without the enzyme, so a massive activation energy. And then the result of using the enzyme is it reduces the activation energy to well under half on this diagram, to enable some of those previously unreacted collisions to react, okay, therefore speeding up the process. Don't worry if you don't understand all of that about activation energy. That is the higher level work. So factors that affect how enzymes work. Extreme temperature, so heat, not low temperature, just high temperature, and extremes of pH can damage the enzymes. We say that the enzymes have denatured. Do not say that they die, okay? Enzymes denature. And basically it means that they change shape, which is irreversible. and the shape of the active site changes. So if the shape of the lock has changed, then the key no longer fits and the enzyme can't do its job anymore. Okay, so it's irreversible, it's extreme hot conditions, extremes of pH, okay, and it changes the shape of the active site irreversibly. So the enzyme can therefore no longer do anything. So let's look at that effect of high temperature, okay? Once the enzyme is heated above 40 degrees, that's when denaturing of the enzyme can occur. So we can see on the picture on the left, the substrate fits in there nicely into the active site, and then it's heated above 40 degrees C, and the shape of it has a kind of elongated, the shape of the active site is no longer a suitable match for the substrate, okay, irreversibly changing the active site, so therefore the enzyme no longer works. So what this is triple science only, this one slide, and it relates to a bit to paper 2 really. So what's actually happened to the structure of the enzyme to cause it to stop working? So this is when it's heated up, okay. The genetic code, the amino acids, the coding of bases in the protein has not changed. But as the protein was made, it kind of folded up on itself, and there were bonds between those amino acids, and they've weakened, okay? So the protein is unfolded, which changes its shape, changes the shape of the active site, okay? That's really high level information there, okay? So don't worry about that too much unless you're looking at triple science. Let's look at a graph, analyse a graph of the effect of temperature on enzyme activity. So in section 1, we've got to think in our chemistry minds now, what happens to particles as they gain energy. Okay, so as particles gain energy, they move around more, therefore they're more likely to collide, and therefore that's why we have the higher rate of reaction there in section 1. Section 2 shows us the optimum temperature, the highest rate of activity. We can read off from the side of the graph, and then as we follow it down we can see that that's at 40 degrees. So we would describe 40 degrees C as the optimum temperature for this enzyme to work. And section 3 is where the denaturing is occurring. So the enzyme's got too hot, the active site is changing shape and therefore no longer working properly. We can look at an experiment for seeing how temperature affects rate of reaction in amylase. And we've done this here with some spotting tiles with iodine. Okay, so we've filled those all with iodine at the beginning. And then we've mixed 5 mils of starch, 1 mil of amylase, and we haven't contaminated the syringes because that could start the reaction before we're ready for it to. And every 20 seconds or so, we've taken some of that mixture, just a drop, out of the beaker and tested it for starch, and that's the iodine test. Okay, so if the iodine stays that orangey colour, all of the starch has been broken down, and if it goes blue-black, there's still some starch there. Okay, so we can see that looking at changing temperatures, we had 10 degrees, 22 degrees and 40 degrees. We can see that there is a much higher rate of reaction at 40 degrees. If we'd gone even warmer than that, we would have seen the amount of time taking much longer because the enzyme would have been denaturing. So that wasn't actually a required practical, but we had a slight twist on that, which I'll talk about shortly, which is how pH affects rate of reaction, and that was the required practical. Okay, let's look at a summary of just bits and pieces that we've talked about enzymes. What is the function of an enzyme? It's to break large insoluble molecules into small soluble molecules. This is so that they can be absorbed through the villi in the small intestine into the blood. What is meant by active site? It's the part of the enzyme that the substrate attaches to. Okay, that's the lock on the enzyme. How do enzymes work? The substrate fits into the active site of the enzyme, it digests it into smaller molecules, and then those smaller molecules are released by the enzyme and the enzyme repeats the process. Where are enzymes made in the body? They're made in the pancreas and the small intestine, that's all of the enzymes, but also some enzymes, protease made in the stomach and amylase made in the salivary glands. What is meant by lock and key? All enzymes' active site is described, an enzyme's active site is described as a lock. It has a specific shape for a specific substrate. Okay, for example, carbohydrates can only go into carbohydrase enzymes. Why do enzymes stop working? They have that irreversible change of the active site due to high temperatures or extremes of pH, where the active site changes shape and the substrate no longer fits. Okay, let's have a look at analysing a graph of pH then. So this is looking at two enzymes. They're both protease enzymes. We've talked about them already, pepsin and trypsin. If we look at the pepsin line first, we can see that optimum level of activity, so the highest rate of activity, has an optimum pH of pH 3. Now in the stomach, we've got the hydrochloric acid, which is pH 2. So that's why pepsin is found in the stomach. It works really well in those acidic conditions. Trypsin is in the small intestine, and we can see from this graph that it has an optimum of about 8.5. Okay, works better in slightly alkali conditions. So the protein goes into the stomach, pepsin breaks it down into peptides, and then trypsin breaks the peptides down into amino acids in the small intestine. What can we say about this graph? We can say that trypsin works over a longer or a wider range of pH than pepsin does. We can say that they both work at pH 5. We can say that trypsin is more efficient than pepsin because it has a higher rate of reaction at its optimum point than pepsin does. And we can say that pepsin works better in acidic conditions and trypsin can actually work best in slightly alkali conditions but can cope with a wider range of pH. Okay, so pH can cause that active site to denature as well as high temperatures. So here we have, this is leading into the pH and amylase required practical. So here we have a rate of reaction versus pH graph for amylase. And we can see, according to this graph, there's quite a range. It works from pH 1 to pH 9. The optimum pH is 6. And we can see that going away from 6 either side towards the 5 and the 7, the activity reduces. So the optimum pH is 6. So for this experiment, we did the same as the temperature experiment. We had the spotting tiles with iodine in, and we added a fixed known volume of starch. We added to that a fixed known volume of amylase, and we had different pHs of amylase. Okay, so we'd added a buffer solution to each of the amylase samples to create different pHs. And then we tested how the pH of the amylase affected the time at which it took for the starch to be broken down. Okay, and we don't know. We could see the graph in the exam in two different ways. So we could see it as it is in this first graph. And this is if we just take pH on the bottom of the graph, time on the side, and a fast or a short time shows a fast rate of reaction. So it actually looks, that top graph looks the kind of mirror image, like it's been flipped over from the rate of reaction graph. Okay, so what we know to be a good pH for the amylase, which was on the previous graph it was 6, but here it looks more like 7. is giving us a very short time. And then what I've done is I've taken that graph and I've just flipped it over like a mirror image, and we can see what looks more familiar to us, which is the rate of reaction graph. Okay, now we can't just flip over a graph. Okay, that was just one way of showing how simply it is to transfer that information. But what we need to do is create a rate of reaction graph. So we look at the total number of seconds that it took to break down the starch. We do 1,000 divided by that time, and it gives us a rate of reaction value that we can then plot, and we will get a graph that looks like the bottom graph. Okay. In chemistry, to work out rate of reaction, we do 1 divided by time. In biology, we do 1,000 divided by time, but you get the same pattern in your graph either way you do it. It's just that you get slightly nicer numbers if you use 1,000 divided by time. Okay, so we may see a graph that has time on the y-axis, and we see this kind of inverted graph. We may see a graph that has rate of reaction on the side, and we see this shape of the graph that we expect to see, that we saw just here. Okay, it might be that we're given the times, we've got to calculate the rate of reaction and then plot our graph for rate of reaction. Okay, let's look at an enzyme summary. Only one type of substrate fits into the active site, like a lock and key. Once in place, the enzyme and substrate bind together, making the enzyme-substrate complex. The reaction takes place and bonds in the substrate are broken. That's what's happening. This long chain molecule is having some of the bonds in that molecule broken, and then the products are released. And it's not that we put in some starch and suddenly it comes out as all this glucose. It may be that the starch goes into the active site of the enzyme and it breaks into 2. So it's now 2 shorter chains of starch, and then one of those goes back in and breaks down a bit more, and now there's some glucose released. It might be that it has to go back in and break the bond in another point of the starch to make it smaller and smaller. So it's not some magical thing where the starch goes in and then it all comes out as glucose. It could be that it takes time. The long chain of starch keeps going into the enzyme at different points to break down different parts of it into glucose. They work best under optimum conditions of pH and temperature, and this is because extreme temperature, so high temperature, and extremes of pH can cause the shape of the active site to denature, to change shape. And enzymes are not used up in reactions, they can be used again and again. We've also got the need to talk about different types of enzymes. Okay, so here again, this is the higher stuff. We know that enzymes are proteins. Ones that break down food used in digestion are called catabolic enzymes. Okay, if you look in the bottom, central bottom of the picture, catabolic enzymes are the ones that break down. And then in plants, they take the glucose and convert it into starch for storage. So they use anabolic enzymes, and these join the molecules back together, making the larger molecules for storage. So taking the small glucose, joining lots of the glucose together and turning it into starch for storage. So there's just 2 types of enzymes: enzymes that break down substances, but also enzymes that build up substances as well. Okay, and I hope you found that useful. Thank you.